The AIRWIC scientific workshop discussion was actually a discussion to dive more into the biology of CLL, but also whether or not there were consequences for therapy. And the talk that I gave was twofold. First was the question, now that we know that we can treat the bulk of the CLL cells, we know that with the new target agents like Venetoclax we can get to MRD negativity, but we still cannot reach to complete clonal eradication. So my first part of my talk was, would it be better to give the two drugs, Venetoclax and Ibrutinib, or a single agent based on biology? And what I actually showed was that both Ibrutinib and Venetoclax resistance highly correlates or corresponds to the increased expression of other anti-apoptotic molecules than BCL2, mostly BCL, XL and MCL1. And then we showed that if you combine the two drugs, that actually is very synergistic, because Ibrutinib chases the cells out of the protective lymph node environment where BCL, XL, MCL1 and other anti-apoptotic molecules actually get increased. And then Venetoclax is much more sensitive for the cells in the peripheral blood. And we showed that that's not only true from a theoretical point of view, but also we had a mouse model where we did adoptive transfer of leukemic cells, T-cell1 leukemic cells, into a wild-type mouse. And we treated the mice with either single agent or double therapy. And indeed we saw a sustained delay of disease progression if you give double treatment. And the next part of my talk was that although you see a significant delay in a recurrence of the disease if you give the two drugs, you still get in all the mice, and also we know in all humans probably, you will get recurrence of the disease. So the question is, should we also work with T cells to eradicate the clone? And then I said a little bit like Asterix and Obelix story, that all over the conference you talk about T cells, but not everywhere. Only in the CLL sessions T cells are never mentioned. And the reason is that they are not functional or dysfunctional in CLL. And then we showed some evidence, very recent evidence, that the T cell dysfunction highly correlates with metabolic dysfunction, and that if you could turn around the metabolic function of the T cells, then actually you can improve T cell function. And of course we don't know exactly how to do that, but that's the part of research that we're currently doing in Amsterdam.